Yeah, yeah. Evening, good morning, good afternoon, wherever you are watching us from. This is microbiology enthusiasts. The first lesson. This is just more of a kind of mentorship program, or just on microbiology. I'll be a presenter today. My name is Martin Masharia. Thank you and welcome. So we'll have our first lesson outline, which is on, first of all, we'll have some introductions and the why. We have a brief introduction to microbiology, and I'll just lay my perspectives. We'll have a day in the life of a clinical microbiologist. Then we have a mention of various equipment, supplies, and reagents that we can use within the microbiology lab. Then we have some closing remarks. Thank you, and please join me along. Yeah, so we'll have some maybe perhaps to introduce ourselves and maybe the why. Maybe it's always a good thing to before you do anything, to just have a why of why you want to do that thing. Yeah, so it's always a nice thing, like why the why should be clear. Yeah, so maybe on my why, I'll say like micro is, although I will introduce it in my next lecture, so in my next lesson. So let's just move along. So a brief introduction to microbiology. So micro is microbiology is derived from two main words. Number one word is the first word is micro, which means small. Then we have bio. Bio means life. Yeah. So then logi is derived from the word logos, meaning study of. So microbiology is a study of small living organisms. So this can be bacteria, viruses, and fungi. Then there has been some debate on whether to include parasites within microbiology. And so you find that parasites, they are also classified as part of microbiology. So some of my perspectives, I'll say that microbiology is it's a sweet fit of MLS. Then it's quite a good avenue to other careers. Like especially you have some interest in research, you like to go into biotechnology companies. Then the other thing is I will say is, microbiology is quite broad, and it's always good to get your poison and run with data, e.g. bacteriology only, because it's quite broad. You see, it has quite some fields of bacteriology. There is virology, there is mycology, there is your poison and run with it. Then it's a bit far from being fully automated, so it's easier to slide through. So we are currently in the era of quite some numerous innovations, especially within the field. So it's always good. It's a nice thing when you are already aware of what micro is, and you have the basics right. Then it's always good to be ready to read a lot, because it involves a lot of reading and keeping yourself updated with the current guidelines, as well as with the current researches, with the current antibiotics that are in development, with the current organisms who has actually changed. So it's always good you must be ready to read a lot. Then microbiology also provides now a room to engage with other cadres. You can engage with, for example, infectious disease specialists. You can engage with infectious diseases pharmacists. You can engage in, form as part of the antimicrobial stewardship committee within the hospital. It also provides a good room also to engage with the infection prevention committee within the hospital. Yeah, so it's a good a place where you can it forms an intertwine between microorganisms and the patient's clinical presentation, and a praise of making now determining the antibiotic that this particular patient is going to use. Then we can separate microbiology with immunology. So immunology is quite a good remains to be the heart of microbiology, because you have to know what are the host defense mechanisms, what is the interactions between the host, between the host, the environment, and as well as, we talk of, when you're talking of microbiology, and especially we are talking of antimicrobial stewardship, we always talk of what we call a one health approach. So a one health approach involves humans, it involves human health, it involves animal health, as well as it involves the environmental health. So let's just have a look at what's the day in the life of a clinical microbiologist entails. Definitely suppose now you have to start your shift. You maybe perhaps receive any handovers that have been there. Then you do some decontamination. This perhaps you brain tease your equipment. Then you do the refilling of your supplies and consumables. The other thing that happens after you're done with all these, you check your catcher reading and reporting. This is always done in the morning, especially now you read the the catchers that has been done maybe a day or 2 days ago. Then you determine on whether they need to proceed for further steps, maybe to do a gram stain, to do identification, and perhaps as well as to the AST. The other thing is follows is routine analysis. Then at some days you might be involved in the EMS committee, attending webinars, section meetings, placing your orders for your consumables and reagents, maybe offering some advice to medical officers and consultants on perhaps which test to request, and maybe some result interpretation, maybe advice to phlebotomists on samples acceptability, the turnaround time for various tests, and maybe at the end of the shift there is always now doing the check, the pending request, and now you issue handovers to the next team. And maybe the last thing that I forgot is as well to do your decontamination at the end of the shift. Then some of the equipment that perhaps you encounter in the microbiology lab, they can be, for example, urine analyzers, blood culture machine, Vitek machine. For molecular, you can have biopharm machines in experts. We have some support equipment like the microscope, the water bath, the incubator, the vortex mixer, the timer, thermometer, Bunsen burner, as well as a biosafety cabin. They quite a number. So it's have quite a number of equipment that we need. Now, it all depends with the complexity of your microbiology laboratory, and as well as with the scope of your tests. Then some of the supplies you might have as slides, cover slips, applicator sticks, the gauze, oil immersion, inoculating loops, your gloves, your face masks. Yeah, so maybe you can think of other supplies that we can have, maybe perhaps within the lab. We can talk of other reagents, other supplies, maybe Whitman, maybe a filter paper, maybe a Bunsen burner. You might have talk of the applicator sticks, the inoculating loops. You might talk of the cover slips. Yeah, so they quite a number. Maybe the staining racks. Those are part of other auxiliary equipment and supplies that you might need. Then some of the reagents. There is one main procedure within a clinical microbiology lab, and number one, the main procedure, the main test that we do is the gram stain procedure. So the gram stain procedure, on the principle of it, it's basically based on differentiating. It's a test that is majorly aimed at differentiating or grouping bacteria based on two staining characteristics, as being gram positive or gram negative. For the gram positive bacteria, they will stain either purple, then the gram negative, they will stain either pink or red, depending on the nature of the counter stain that is used. So some of the gram stain reagents that we can use, we can have a crystal violet, glucose iodine, acetone, safranin, or stroke neutral red. Then we have Zn is another test that is used for Mycobacterium tuberculosis. We can use carbofuchsin, acid alcohol, methylene blue, or malachite green as your counter stain. So we have quite a number of reagents that you can have, and all your reagents, they are based on the test that you want to use to perform. Like for example, the oxidase reagent is a reagent that is majorly used for oxidase test. Can have normal saline, Indian ink for Cryptococcus neoformans. We have potassium hydroxide for fungal staining, hydrogen peroxide for Attescott catalase, coagulase plasma for Attescott coagulase. Then you can have our culture media as well. So then within microbiology, you find like, as I had said, that immunology forms part of, it's the heart of microbiology. So you find like, yeah, this is what I had said. So immunology is the heart of microbiology. So you find like within the microbiology section, you find like serology forms part of microbiology, and serology is majorly involved with the antigen antibody reactions. So some of the rapid kits perhaps you can have, maybe you can list them in the chat. Maybe you can have, for example, a H. pylori, Helicobacter pylori antigen test, Salmonella antigen test kit. You can have a TET for dengue fever. You can have a pregnancy detection test kit. You can have a test for example, Clostridium difficile, Campylobacter coli. You can have a test for Vibrio cholerae, cryptococcal antigen. Yeah, so it all depends with the various types of serological tests that you want to support perhaps your cultures. Yeah, so that forms the part of our lesson one of introduction to clinical microbiology. So in our next lesson, we'll have a brief breakdown on the various microbiology sections. So and these sections will majorly emanate from these divisions of microbiology as bacteriology, virology, mycology, as well as parasitology. So thank you so much. And in case of any question, please feel free to post your questions in the chat. Thank you. And my name is Martin.